Hi, everyone. So we're going to start the second session of the NetPyNE tutorial. This is part of the CNS 2022 satellite tutorials. I'm not sure if there's anyone new here. If so, please let me know in the chat. But essentially, you can download the slides that we're using. Here's the link. Right here at the top. I'm going to paste it again in the chat. Okay. And we're going to start with session 2, which is focused, let me find it here, focused on the Jupyter Notebook programmatic version of NetPyNE, so where we're using Python code. And here we're also going to use the open source Brain platform, and in particular the JupyterLab tool that it has built in. Okay, so let's go ahead and well, quickly an overview of what we're going to see. So I'm going to go over this JupyterLab environment in open source Brain. Then we're going to see the network oscillations tutorial, the same one that we saw in the graphical interface, but we're going to play around with it programmatically, which I think is useful, so you can see the pros and cons of each, and also understand how it's implemented at a programmatic level, now that you understand the different components of the model. Then we're going to see a brief overview of the NetPyNE data structures and how to access the different components, and also how to import different cell models, and some short tutorials on the different analysis and plotting functions that we have. And we're going to finish off with network parameter exploration, again, similar to how we did it with the graphical web application, but all doing it directly from the JupyterLab, so using the terminal and running Python scripts. Okay, so in general, just to mention that, of course, this programmatic approach gives you some more flexibility to what you can do, but it also loses the sort of intuitivity on the ease of use that the graphical interface provides. Okay, so let's go ahead, and the first thing we're going to do is just play around a bit with the open source Brain JupyterLab environment. So if you have still open this GUI that we had before, you can switch to the JupyterLab environment just by going to the 3 dots here at the top left and clicking on Open with JupyterLab. And so this is going to open the same workspace that we are using right now, but in the JupyterLab environment. You can click and do that straight away. You might need to log in again. If you're starting from scratch, let me just show you. So you will, after you log in, you will see here your own workspaces. Let's reload. Okay, so this is the workspace that I was working on before for the morning session. So I'm just gonna open the same workspace, but instead of using NetPyNE, which is the NetPyNE web application, I'm gonna use JupyterLab. Okay. If I click here, I'm gonna get this same screen. Okay, again, some of these, the main points will be included in the slide, so you can follow along, different steps to follow and so on. But stop me anytime if you have any questions or are falling behind. Okay, so the JupyterLab basically has these 2 main windows or components. Here on the left, you have a file browser with all the files that are accessible in this workspace, and on the right-hand side, you have this viewer of the different files that are open or terminals that you're using. So initially you get this launcher, which allows you to create either Jupyter notebooks or open a console or a terminal and so on. And so the first thing that we're gonna do is just explore the experiment that we launched before from the graphical interface, which was this exploration of parameters in the oscillatory network. And we looked at one of the parameters, the inhibitory synapse tau. So we're gonna see the files that were generated by that experiment directly here in the JupyterLab environment. So this experiment will be found in the experiments folder, and it will have the name, the folder name will be that of the experiment that we labeled before. So in this case, OS for oscillation. And here inside, you can see all the different files that were generated. So we basically have 3 sets of files, one for each of the parameters that we explored. So for each simulation that was run, we have essentially 4 files. This config.json is the simulation configuration, and JupyterLab has this nice feature to visualize JSON files in a very clear way. So here we have all the properties of the simulation configuration. That's a component of NetPyNE. And then you have the JSON file with the output data. And this output data file has different elements. So first you see net, and net has the net params and the cells and populations. So params will contain essentially the network parameters, so what we were defining from the graphical interface. So things like the cell parameters, where you can see, for example, the pyramidal cell and the sections, the soma, geometry, and so on, in a very nice hierarchical organization. You also have, for example, the connectivity params here, con params. We have this connectivity rule E to I with all the properties. Inside net, you also have the cells of the network. So in this case, there were 40 cells, and for each one you can see the properties of the cell, the sections that it has, again, the soma, mechanisms, and so on. And you have the output simulation data, sim data. So here we have, for each of the simulations, the voltage of the soma, which was recorded only for one of the cells. You have the spiking times. This is a spike T is the spike times, and a spike ID tells you the ID or the global identifier of the cell that spiked. So for example, the first spike happened at 8 milliseconds and was fired by cell number 12. And in this case, we also have the dipole currents, which we were trying to use to plot the EEG signal. And these are calculated for each of the cells in the network. And this is a 3-dimensional vector for each of the cells. Oh, this is actually, sorry, this is over time, yes. Yeah, so this is for 1 millisecond, sorry, for 1 second of simulation. We have 10,000 steps, and each one includes the dipole current. Okay, and you see we have that for each of the 3 parameter values we explored. Sorry, and we also have this run file, which gives you the output of the simulation. And you see that it specifies the network was created with 2 cell populations, the number of connections, the number of spikes, for example, and so on. So we see the number of spikes here for the first simulation was 1,000, an average firing rate of 27. For the second simulation was 14 hertz on average. And for the last one, which if you remember, died off pretty quickly, the firing rate was only 2 hertz. Okay, so this was just to illustrate that you can see things that happen in the graphical interface have an effect on the workspace. And for example, here we're generating output files for each of the simulations that are executed. Any questions? Okay. So let's move on to the next section. Okay, so now we're gonna use the JupyterLab to run the same tutorial that we run using the graphical interface, tutorial 3. And we're gonna use a terminal, normal terminal. So we're not gonna use a Jupyter notebook, but just the regular Python files and a terminal to run with Python this tutorial. So let's go ahead. So these tutorials are gonna be found in the folder called NetPyNE tutorials. And then inside that folder, we have NetPyNE workspace master. So this is where these tutorials live. And here we find a bunch of files already. So just so you know, the tutorials in the graphical interface are basically copied from or instantiated based on this data. So they appear, the fields in the graphical interface are filled in automatically from these files, from these Python files. So in particular, we're gonna look at this GUI tutorial, no RxD. So we're gonna omit the reaction diffusion part just to make it a bit faster. So if we open the file, we're gonna see for the first time sort of NetPyNE model defined in a Python file. And again, this is exactly the same thing that is loaded into the graphical interface, and it's just filled in within the different panels of the graphical interface. So let me briefly go over the components of this model. Okay, so let me close some of the windows here. And the first thing we see at the top here is this line from NetPyNE import specs. Specs is the sub package that allows you to define the specifications of the network model. So the first component we need to build is called NetParams. This is an object, and it's going to be the top level object that contains everything. And it's of the type NetParams, of the class NetParams. So inside NetParams, we're going to have some properties that are sort of the baseline level, which include the size of the network in the 3 dimensions, as we saw before also in the GUI. And we can also define a couple of properties that are used for connectivity. And I mentioned this before, things like the propagation velocity and the probabilistic length constant. Okay. Next step is, as in the same fashion as in the web application, we're going to define the cell properties or the cell parameters. Actually, it's maybe useful if I open at the same time the graphical interface to show the comparison of how you would do it graphically, since you already played around with it. Okay, so the cell parameters are the first component, and similarly to what we did in the GUI, we're going to import those from existing cell models. In this case, we're actually loading from this JSON file, which contains the parameters of a cell that was previously saved in NetPyNE format. And for the inhibitory cell, we're loading directly from a HOPE file. Notice the flexibility. You can load from JSON, from HOPE, from Python files, and basically NetPyNE does all the job behind the scenes to fill in the parameters of these cell types. Okay, so here we have the same. Let me load tutorial 3. Okay, and yeah, so we see here the E and I cell types. and the cell types component. Now we move on to populations, and we will see the same thing here in the script. So we have 6 populations, and notice we are now filling in these pop params, which is a component of the NetParams object. And so we fill in with the 6 different populations, E2, I2, E4, I4, E5, I5. And for each one, we specify the cell type, the number of cells, and the Y range. So this is the cortical depth range of values for each of these populations. For layer 2, we select between 50 and 150 micrometers, for example. Next component is the synapses, and we fill in this scene mech params component. We have 2, excitatory and inhibitory, and for each one we define its properties. The main difference, of course, being the inhibitory has this reversal potential of -75. Okay, and we can see the same here, excitatory and inhibitory. Okay, so moving on, the next component of this model is the stimulation. So we want to stimulate the network to generate a background stimulation, in this case using a NetStim that's going to fire at 20 Hertz. And then the way this works in NetPyNE is once you have a source of stimulation.
Salvador,
yeah.
I've got a question about the synaptic mechanisms.
Yes.
So if you wanted to do an NMDA conductance, for example,
yep.
How would you define it there? Because these are simple ampere and GABA.
Yes, well, the NMDA you can define in multiple ways. So you can simply use this double exponential and just make, for example, these constants, time constants longer, which is typically what you see in NMDA. But of course, you can also use a separate mod file. So this mod entry here is basically pointing to the mod file that you're using. Mm-hmm. And so if you have compiled different mod files that are specific for NMDA, you can just select that value here, the name of the mod file, and then fill in any parameters that are specific for that mod file.
So that mod file would just have additional information about the voltage dependence of the conductance, for example?
For example, yes. Okay. And can potentially have many different parameters as well to customize.
And so any mod file that's available basically is compatible with this, and you can just import that in at any time.
Yes.
Yeah, great.
Correct, yes. Okay. If they're compiled at the beginning before running the simulation, then you can use them straight away. There's weird cases for some mod files that are more complicated because they have these pointers and so on. For example, for gap junctions. And in those cases, we've made sort of special treatments within NetPyNE, but those are very rare cases.
Okay, great, thanks.
Thank you. Okay, so yes, we're talking now, discussing the stimulation. So notice how we first create the source of stimulation. In this case, this background of, it's a NetStim with 20 Hertz noise, 20 Hertz rate and 3.3 noise. And then we tell NetPyNE, and we can structure this maybe a bit more clearly in this way. The different fields of this target. Okay. So notice this stim target params object here. We're calling this entry background to E. So what we want to do is target all excitatory cells with this background source of stimulation. To do that, we say the source is background, which we defined here. The conditions of the postsynaptic cells that are going to be target are that the cell type has to have the property of being E. So all cells that have a cell type of type excitatory will be targeted in this case. The weight of the connection is going to be 0.02. The target section, the soma, and the delay is going to have this expression as we saw before. So it's a normal distribution with mean 5 milliseconds. And so you're going to have some variability also in the delays. And the synapses we're going to use is this excitatory synapse. Okay. Same thing for the for the inhibitory synapse, for the inhibitory background to inhibitory stimulation with some slightly different properties. Okay. And the last component that we're going to define for the network is the cell connectivity rules. So here, as we saw before, we're going to have, let me just reformat this properly. We're going to have two connectivity rules, one that connects excitatory cells to all other cells, and one that connects inhibitory to excitatory cells. And we already saw before in the graphical interface, the resulting connectivity matrix and how these very short, simple connectivity rules could result in quite a complex connectivity distribution. So just briefly here, for example, we specify the conditions of the presynaptic cells, which is all cells with cell type E, the conditions of the postsynaptic cells, all cells within a cortical depth of 50 to 500 micrometers, probability of connection of 0.1, weight that depends on the postsynaptic cell cortical depth, and a delay that depends on the distance between the cells divided by the propagation velocity. Similar thing for this other connectivity rule, we define all the properties. And now we are done defining the network parameters. The next step is to define what we call the simulation configuration, which are things specific to the simulation itself, things like the duration, the integration step, for example, the recording step, the name of the output file. Here we have a bunch of traces that we want to record from. Yes, Angus?
Yeah, so in terms of is there any help on the integration step size? Because obviously that can be quite critical in simulations and in terms of stability, but also, well, and it depends on the numerical integration method. Yes.
This is, in the same way as Neuron is, flexible in terms of what the user defines. There's no constraints here. You basically need to know what you're doing in terms of computational neuroscience and modeling. In here, we're using this value 0.1 millisecond, which is slightly high and can give some issues for some networks. But in this case, because it was a simple toy model, we just wanted to run faster. The default typical value can be 0.025, for example. But again, there's nothing specific in NetPyNE that tells you what to use. You just need to learn these methods and maybe base it on existing models and so on.
In terms of building with the graphical interface, presumably there's some sort of default.
Yes.
Is it flagged that you need to think about this property carefully, or anything like that?
Yes, that's a good point. So all the properties, the simulation configuration properties have default values, and you can look at them in the reference here. Simulation configuration. Here you see the DT integration step. The default value is 0.025. So if you leave everything blank and you don't fill anything, you will have reasonable values.
Mm-hmm. Okay. Thanks.
Thank you. Okay, so yeah, finishing off with the simulation configuration, we set up some traces that we want to record. And there's some also nice documentation on how to specify this. Here we're recording the voltage in the soma, this potassium current, the intracellular concentration of calcium, and the extracellular concentration of calcium. Next, we decide here that we want to record local field potentials. And for this, what we do is essentially define a list of 3D points where you want to place the LFP electrodes. So here we're using this sort of list comprehension to make it a bit more generic, but this would look potentially something like a list of 3D points. So 555 would be one location, for example, and 100, 100, 100 might be a second location. So this would place 2 LFP electrodes at these 2 3D locations. Okay. And finally, we're going to tell NetPyNE what plots you want to show after the simulation is complete. You can also then, once the simulation finishes, you can run your own plots and analysis manually from the terminal, or if you're in a Jupyter notebook, from the Jupyter notebook. But you can also tell beforehand, if you know some plots that you're going to want for sure, you can specify them here, and this would be plotted. You can also select to either show them on screen or save them as figures. And you can customize what plots, features you want to show, and so on. Okay. And just notice before we run the model, in this case, because this is the code that is being used by the graphical interface, we have to add these 2 lines here that essentially make it easier for the user to access the model afterwards. But since we're gonna now we're gonna run it directly from the Jupyter notebook here from the terminal, we're gonna remove these 2 lines. You can either comment them out or remove them. And we need to uncomment the last 2 lines. And these last 2 lines, if you notice, are the only ones that actually get executed, this last line. All the previous code here was filling in dictionaries, essentially, dictionaries and lists. So there's nothing that's really being executed. And the only execution line is this one at the end, where you create the network, run the simulation, and perform any analysis you've specified. Okay, so please, if you're following along, remove these 3 last lines and uncomment these 2 lines to actually create and simulate the network here. I think this is also included in the slides. Yeah. So the step number 5 is to remove these lines that include the NetPyNE Gepetto and uncomment the last 2 lines to run the simulation. So this is the general structure of all NetPyNE models. So all NetPyNE models that you look at will have the same filling in of parameters, of the net parameters, cells, population, synapses, stimulation, connectivity, and then the simulation configuration with just a single line at the end to run the simulation. This can be potentially split into separate files, but the overall structure will be identical, which makes it easy to follow along and understand the different models. Okay, so now we're gonna go ahead and actually run this model. So we open a new, we go to File, New, Terminal, and just remind myself. Okay, so we first have to navigate to where the model is located. So we go to NetPyNE Tutorials, CD NetPyNE Tutorials. Inside here, we go to NetPyNE Workspace Master. And here is the model already. So we can just run IPython -i to make it interactive, so we can plot more things afterwards. And the name of the file, which is GUI tut3 no RxD. Okay, so this is also on the slides, so you can copy and paste. Important, we forgot to compile the mod files. So this is something that you have to do for any neuron models that have external mod files. So the mod files are the files that define different ionic channels, synapses, and essentially any mechanism that is used in the cell. So we can, to compile, we just have to type neuron IV model and the location of the mod files, which in this case is mod. It's a folder that contains all the mod files. Okay, that compiles all the mod files. You can have a look also here in the file browser. You go inside mod, you will see all this list. Most of these are channels. Okay, let me add this to the slides. Okay, here, compile mod files. And now I will run the model again. Okay, so this created the network with six populations, 60 neurons in total, 300 connections. It tells you the time it took, 6 seconds to run the simulation. And we got a total of 1,600 spikes. And that's it. So now it shows here all the different plots that were generated, and most of them are set up to be saved to file. So we can now navigate to the same folder, and you see these different plots were generated seconds ago. Let me highlight here that we selected to plot what we call interactive plots, which have an eye in front of the plot. So for example, we have both plot raster and eye plot raster. They're both valid, but if you put an eye in front, it means it's interactive, and it's plotting using a package called bokeh. And this is a package that then generates the plot in a way that it's interactive. So if I click here in the raster, for example, and we have to say trust HTML to actually visualize it. So you see now the raster plot, and this is interactive in the sense that you can move it around, you can hover over different spikes, and it tells you the spike time and the cell that spiked, and so on. So this is one of the plots that was generated. We also have the LFP signal, same as we saw before, and the power spectral density of the local field potential, and the spectrogram. Okay, so this, yeah, we've basically seen how to run the same tutorial that we saw before, which is tutorial 3 here, both from the graphical interface and getting the same results directly using terminal and simple Python scripts. Any questions? Okay, so let's move on then. Done this. Okay, so now we're going to see another feature of this JupyterLab environment, which is the ability to add resources. Resources meaning mainly files from different repositories. So to do that, let me just close all these files. And so to do this, we're going to open this tab here in the left. And we get this link now to add a resource. So we can click on add resource. And we have two options. One is by URL or from an OSB repository. So Open Source Brain already has a number of built-in repositories that you can clone. So we see here the Neuron course 2022, which I assume was for the course yesterday. We see some from Arbor, the Neuron Simulator. We also have, or we should have, oh no, NetPyNE is not here. But yeah, it will be added at some point. And let me see if we can actually find it. NetPyNE. No, this one is not here. But what we're gonna do now is just add a repository from GitHub. So this shows you how to get any GitHub repository that you have with any model you're building and upload it so it's available in this JupyterLab environment. So I'm gonna go to GitHub. I have it here open already. So the repository I want to clone is this one that's called eBrain CLS Interactive, which is a number of Jupyter notebooks that were created to run on the eBrains platform. And we're going to now run them here on the open source Brain platform. So what I want to do is copy the link of the zip file that contains the repository. So to do that, you can go here in the code, click on code, and then where it says download zip, you click on the right-hand side or the right bottom of the mouse and say copy link address. So this is also shown on the slides. You can go directly here. I'm going to paste this link on the chat. That's the repository we want to clone, and you have to copy the link to the zip file. I'm actually going to paste also that link there so you can use it straight away. So this is the link to the zip file that you can use directly on OSB. Okay, so just to recap, we're going to try to add a resource here in our JupyterLab environment. So we click on add resource and we're going to do it by URL. So here where it says paste your URL, we're going to paste this zip file link. We can also give it the resource a name, and I think it's going to be useful if it's a bit more informative. So we're going to call it, for example, Jupyter Notebook Tutorials, or anything that works for you. Okay, any questions? Okay, so let's go ahead and upload this. So now if you go back to the root folder of your workspace, you should see this new folder being created, Jupyter Notebook Tutorials, or whatever you called it. Okay, and here it is. So we have to navigate inside. There's another folder called NetPyNE Tutorials Master, and inside here we will see. Oh, I don't know what happened here. Let me just reload. Actually, maybe this is a good chance to show this other feature. I'm going to show going to the last slide. So there's a way to know how many sessions you have open. And since we've been opening and closing and maybe had some issues, you might have several of them. So you can either click in this link, which is in the last slide of the slide deck, or from the JupyterLab, if you go to File, Hub, Control Panel, you will also get the same window. So if I click on this, it tells you the list of sessions that you have open. I currently have 2, so that's correct. But if you have more than you want, you can close them by clicking here on Stop and then Delete. Okay, so let me go back here. Not sure exactly what happened here, so I'm gonna try to go back to the main screen and just open this workspace again using JupyterLab. Okay, so here we are. So yeah, so I have this resource that I cloned with a bunch of Jupyter notebooks that we're gonna use for the remaining of the tutorial. Please let me know in the chat if you've had any issue with this, because we need these Jupyter notebooks for the remaining. So if you have any issues, let me know in the chat. Or you can also unmute. We're just a few now, we're 13, so this is more a close family reunion. Okay, so I will assume that you've all managed to get these files cloned to add this resource, this GitHub repo. Actually, let's do it the other way. Can you please say in the chat if you have these files already added to your workspace? Okay, thanks, Marinus. Yep, great. 1, 2, 3, 4, 5, 6. Okay, it's probably most people. Okay, the remaining who didn't answer, I might be just not here. But if you are following along and you have any issues, please let us know. Okay, so let's continue. So what we're gonna do now is run the oscillations tutorial, which we also did from the graphical interface. And we're gonna do this using a Jupyter notebook. We're also gonna play with some of the values as we did before and try to change the oscillation frequency. So I'm gonna open the one that says NetPyNE tutorial 3 oscillation. Notice that before we ran also another model that we did before this morning, but using a Python script and using the terminal. And now we're gonna run it using the Jupyter notebook, which is kind of more interactive. Okay, so let's have a quick look at the model definition here. So we have a bunch of initial lines that we require, importing or installing Neuron and NetPyNE, and these lines which are required to plot plots in line so that you can see them within the Jupyter notebook. And then we have the definition of the network parameters. So as before, we create this netparams object. We create a simple cell type. Notice that here we're not importing, we're just defining the parameters straight away in this very simple hierarchical manner. So you have inside the cell type peer, you have sections, you have a soma. The soma has a geometry with 3 parameters. The soma also has some mechanisms. In this case, just the Hodgkin-Huxley default mechanism, which has a sodium, potassium, and leak channel. And that's all you need to define this cell model. We then create 2 populations, the excitatory and inhibitory populations, both of the cell type pyramidal, because this is a very simple model. We create 2 synapses, excitatory and inhibitory. And again, we create background stimulation, an ETSIM of 50 Hertz, and target all the excitatory population only in this case. And here, because we want to have oscillations again, we have these 2 simple connectivity rules, connecting the excitatory to inhibitory and inhibitory to excitatory. And this creates these recurrent connections that result typically in oscillations. Finally, we have the simulation configuration, as we saw before, with some typically used parameters and some of the plots that we want to show. And at the end, we just create, simulate, and analyze. So let's go ahead and run this Jupyter notebook. So the easiest way, just to click here at the top on the double triangle, which is clear, and run everything. So here we're also going to try to plot the EEG signal. Let's see if we have now enough memory. So to do that, the only extra line that we needed is this record dipole equal true. So this tells NetPyNE that we want to record these current dipoles for each of the cells, and then that allows us to separately plot the EEG signal. Okay, so we're already seeing the output of the simulation. We see that the two networks, two populations were created. And we see this LFP. And actually the dipole and the EEG did work. So it generated these two figures here. You see they were created seconds ago. So we can open them. I'm not sure why they're not showing here. So we can try something like show fig equal true. See if that helps. And perhaps this matplotlib inline, which is sometimes required. Well, maybe it's, yes, it also might be a memory issue and it's not showing within the Jupyter notebook, but it was generated and we can open it here. Oh, here it is. Okay, great. So yeah, we can see here now, this is the EEG plot and it's showing several things. So we, as I mentioned before, this is locating EEG electrodes all around a head volume model of a human head. So all these different locations are the locations of the electrodes, and the color represents the amplitude of the EEG signal recorded at each location. We are assuming that the actual tissue being modeled is located at this star. All this can be, of course, customized. And then we have here the EEG signal recorded at all the different electrodes, and the average, which I think is in green, and it's overlaid. Okay. Okay, and there was a small issue here because we wanted to plot a bunch of plots here that I specified, but they did not show within this simulation. And that's because sometimes you need to modify this to specify the matplotlib inline right before the plotting. So I'm gonna do a quick trick here, which is to first create the network, then simulate it, then call this matplotlib inline, and then plot. This also shows you that you can break apart the different steps of the workflow. So you can create the network separately, then run the simulation, and then analyze. And this is also useful because here you can potentially interact with the network that has been created. So you can do things like sim.net, .cells, zeros, sections, soma, and so on. And we will see a bit more about this later on. You can change properties and basically modify the network as you will. Okay, so let's try to see if this works. I think I mentioned this in the slides because it happened before. Yes, so you can make these small changes in your own version, which is just splitting apart the create, simulate, analyze call. I can also copy the code here in the chat in case that's easier. Chat. Okay. So now if I run this cell, notice how the other plots, some of the other plots did show because they were called using separate function calls after the whole simulation had completed. And now we are also getting the ones that are specified beforehand. through this simconfig, they are showing here now. Okay, so we have these voltage traces, the raster plot, a spike histogram, and we have this nice, this is a spectrogram of the spiking of the network. So basically bins the spike times and shows the spectrogram. So it's a very nice, clear way of knowing what is the oscillation frequency of the network. And we see very clearly here, it's around 37, 38 hertz, something like that. Okay, so let me just catch up with you guys, because now I'm going to ask you to do a small exercise. So let me know if you have everything working, if you've had any issues. Okay, so if you go to the bottom of this Jupyter notebook, you will see here the exercise laid out. And it's basically starting from this Jupyter notebook that we just run. You need to modify some parameters to get a different oscillation frequency. So we basically want to change this line here that's currently at 37, 38 hertz and move it around, either make the networks oscillate faster or slower. And as mentioned here, you can change any type of parameters from the connection weights, probabilities, delays, biophysics of the cells, anything, and just rerun again and see what happens. A quick hint, just to make it faster to run the simulation, you can comment out these two lines. So we don't need to record LFPs, and we don't need to record these dipoles. And now you can just run this cell, which you can do by pressing Shift-Enter, and see the output directly here, pretty fast. Okay. And so you can directly change any parameter. So for example, I'm going to change this probability. I run again, and I see directly the output in a very fast way. Okay. So this already changed the oscillation. It actually removed completely the oscillation. And now there's a lot of noise in the network. Let me put the original value back, 2.7. Thank you, Angus.
Thanks very much.
See you. Thanks so much. See you. So for the rest of the people, I'm going to give you 4 or 5 minutes just for you to play around with this exercise, which again is explained at the end and consists of you trying out different parameters, changing different values, and see if you can change the oscillation frequency of this network. Next, I'll ask a couple of you to maybe give some suggestions and see what parameters work for you. Okay. By the way, since we're only a few people, if anyone wants to turn on the camera, you're more than welcome. It's always nice to see human beings, but you're also free to stay as you are. Okay, so I'll give you a couple more minutes just to play around with this. As a reminder, you just need to change parameters here for connectivity or biophysics or whatever you want, and just rerun this cell, and then you will see the results. I'll be back in a minute. I need to get the charger for the MacBook. 1 second. Okay, so here we go. Please, can you post any solutions you found in the chat, and I will try them out. So specify the parameter that you changed and the value that you used. Okay, thanks, Marinos. So I2E probability, you reduced that to 0.07. Okay, I'm gonna try that and run. Okay, and what happens here is basically any oscillations are completely destroyed. The cells are firing at quite a high rate, and there is no sort of synchrony between the populations. They're not synchronized, so you don't really see these oscillations, and yeah, the peak frequency here disappears. So yeah, good. Thank you. Let's put back the value, and thanks, Katarina. I2E weight 0.5. 0.5. Okay, let me try this. Okay, nice. So here we're actually seeing more synchrony than before. And we see a very clear change in oscillation frequency. Now it's down to 22, 23 hertz or so from around 38. So very nice solution, thanks. And Carmen, I to E delay equal 50, let me see. So I to E delay 50. Let's run that. Yes. Okay, so what happened here? We're also seeing, similar as before, the sort of synchrony between the populations is no longer there. So the eye cells are firing quite high, and you just get these very high noisy frequencies here in the gamma range, but any oscillation frequency has disappeared here. Okay, so Evan says, let me put this back. This was 5. The tau 2 of the inhibitory from 5 to 2.0. Right. Okay, and we see, nice. We see a change, and the peak frequency has now gone up to around 60 hertz, which is interesting because it kind of oscillates, goes up and down between 50 and 55 hertz maybe. So that's a nice, interesting one. And let's try just one more maybe. The I2E weight 2. I2E weight. Oh, I think we tried a similar one before. No, let's try, see what happens. Okay. Oh, nice. So yeah, we get very high synchrony. We can see here the synchrony level 0.82. And we get a very nice peak at 20 hertz, very strongly marked. And some gamma here, very nice. Okay, fantastic. Thanks everyone for playing around and providing the solutions. And hopefully you've seen how easy it is to play around with the network model and see the results using these Jupyter notebooks. So let me know again if you have any questions, and if not, we're gonna move ahead and go to the next section. Okay, and this section is the NetPyNE data structures. And for this, we actually can use the same Jupyter notebook we are playing around right now. And what we're gonna do is just explore how the internal structure of these models looks like. We're not really gonna modify anything, but just navigate through it and explore what the components are. So let me start with this overview diagram. And here we're seeing how the network is structured internally. So the top level, we have net. And this net can be accessed directly. I'm gonna go to the bottom here and just create here a new cell. So you click the plus, you can create a new cell. And here at the bottom, you can play around. So if I type sim.net, this is the top level. And it's what we're seeing here at the root. And as you can see here, it's an object of the type network, of the class network. So if I go inside, we're gonna see two things. We have the populations and the cells. So I can do sim.net pops. And we can see that we have two populations, the E excitatory and the I. Each of them are of the type pop. And I can do sim.net cells. And this gives me a list, as you can see specified here. Cells is a list Each of them is an object of the type compart cell, compartmental cells. So these are multi-compartment cells, basically. Okay, so what I can do now is go inside one of these cells and explore what it has. So let's briefly look here. As you see, as you move to the right, you're going sort of deeper and deeper into smaller scales. So here we are at the population level, then at the cell level, then sort of the morphology and the biophysics. So if I go inside one of the cells, I have, for example, the tags, which gives me information about this cell. For example, the population, the cell type, the location. And then I also have the sections, which is a list of all the different sections contained within the cell, actually a dictionary. I also have the connections of the cells, and I also have the stimulations that are targeting the cells. So this is quite useful if you want to do any kind of analysis of your network connectivity or of properties. You can access all this information here. And let's look as an example at cell 0. If I plot the tags, we see that it tells me it's of type pyramidal, it belongs to the population E. These are the XYZ locations in micrometers, and this is the same but normalized between 0 and 1. I can also say sex for sections, and here I get this dictionary. If this is complicated to follow, you can say keys, which is a basic command in Python. Oh, this is not what I expected. Well, you have all these internal Python components, but basically at the beginning you see here the soma. So this is telling you that this cell only has one section, which is the soma. So we can now go inside the soma. Notice this is now a dictionary, soma. And this gives you all the information about the soma, from the mechanisms, you have the morphology, and so on. Some interesting feature of NetPyNE is that you can navigate this both using a dictionary format or what we call a dot notation format. So you can do the same here and type soma, and you get the same output. So for example, you could do soma.mechanisms.hh and see all the properties of these Hodgkin-Huxley parameters. Okay. Any questions here? Okay, so this is basically what I was explaining here. And these are examples of the different things you can look at. Let's look at some of the connections just to see how they're structured. So if we go in this cell and we type cons, we see the list of connections. These are all the connections targeting this cell. So this is the postsynaptic cell. Connections are always stored on the postsynaptic cell. This is due to how a neuron works. So we see we have around 20 connections or so, and you can see the properties of each one. You can also look at the stimulation, stims. Okay. And I think that's it at the cell of at the level of the cell. So something important is that this is all Python structure, but within this Python structure, you also have access to the neuron objects. So the neuron objects are what is required to be created in order to run the simulation neuron. So for example, for the cell, the section soma, there is one object here or one field called H object. This is always the name of the dictionary entry. When you want to access the corresponding neuron object, you type H object. This is a bit more advanced, but if you've played around before in neuron or you've used neuron to simulate models, then this is what you're used to. And here you can do things like the length of this section, for example, and it tells you it's 18.8. So this is a way to access the actual low-level neuron objects that are being used. Okay, and next I wanted to show the simulation output and how that's stored. So this is a different structure that you can access via sim.sim data. Okay, and here in this case, we have several entries. As we saw before, we have the spike times, we have the spike IDs, which are the cells for each spike time, the corresponding cells that are firing, and we have the voltage soma stored for one cell in particular here, and we have a time vector. So for example, I can do sim.sim data, spike times. This is stored in the NetPyNE, in the neuron format, which is a type of vector. But if we just want to visualize it in Python, we can just surround it by list, and then we get the list of spike times. And of course, this is very useful to do any analysis that you want to do afterwards. You can use this. And the same, for example, for the voltage soma, you would do something like this. Each cell is stored separately in a dictionary. Cell 1. And we want to have a list, a Python list. Okay, and this is the voltage. Here you would also have the LFPs if they're recorded, and these dipole moments if they're recorded. So I think that concludes the NetPyNE data structures. There's more documentation on the website that describes all these components. And there's also a tutorial that I mentioned here in the slides. It's tutorial 7. One question. Okay, great, thank you. Tutorial 7 shows how to modify the network after it's been instantiated and before simulating. So this is useful for many different things, particularly when you get more advanced and you want to customize aspects of the network. So you can do tutorial 7, here is the link, and follow through, and it shows how to change cell properties as well as connectivity. Let me know if you have any questions. And if not, we can move along. So it's 12:30. Maybe we can do a short break. 1:30. Yeah, let's do maybe just a 5-minute break before we finish off the last part of the tutorial. So next we're gonna see how to import different cell models and how to do some analysis and plotting, and Joe will lead these 2 sessions. And finally, we're gonna see how to do network parameter exploration using the programmatic approach. Okay, so we'll see you back in 5 minutes. It's now 1:45, so we'll see you back at 1:50 in New York Eastern Time. Okay, so yeah, this was a short 5-minute coffee break, and we'll continue now. And Joe, please. Thank you.
All right, hello everybody. Let me share my screen here. Where am I? All right. Share screen, continue. And yeah, that one. All right, looks like I'm sharing my screen. So yeah, so we're gonna do a couple tutorials now that people ask for a lot. So the first one will be importing external cell models into NetPyNE. And then after that, we'll look at some of the analysis and plotting stuff that we can do. But yeah, so you should have already gotten the zip from the GitHub repo and loaded it into your open source brain. So if you still need to do that, raise your hand or let me know. Otherwise, I'm going to assume you've already done it and carry on here. So yeah, so for this one, we are going to use this NetPyNE 2.2 importing cell models. This notebook here. So I'm going to double click on that. Okay, and it opens up a Jupyter notebook here in open source brain. We do not need to change the kernel. I guess we need to update this tutorial, but yeah, we don't need to change the kernel anymore. But yeah, NetPyNE allows you to import cell models previously defined from external files using this import cell params method. And we're going to go through a few different file types right now using this and show you how that's done. Yeah, so basically for this, you're going to need a label for your cell type, conditions, file name, cell name. Anyways, we'll get there in a second. There is this import synmex option also. So if your cell model includes synaptic mechanisms, you'll need to set import synmex equal to true. And yeah, so as we've been saying, there's a lot of people have made a lot of different models over the years. A lot of those are available at ModelDB, among other places. But yeah, so it's a great place to start modeling is to use an existing model, get it running yourself, and then play around with it, improve it, see what you can do. So yeah, so let's go ahead and get started here. We're going to install Neuron and NetPyNE, and we're going to do this matplotlib inline command so we can see the plots. And in a Jupyter notebook, you can't just hit enter, it'll add a line. If you want to run something, you have to hold down shift and then hit enter. And that's going to run these commands here. And yeah, the requirements are already satisfied and it's done. Okay, so everything is installed. We are going to clone a GitHub repository that has these cells in the external files. So if you want to run a command in the terminal from a Jupyter notebook, you have to have this exclamation point in front to let it know that it's a terminal command. So I'm going to shift enter there, and that's going to clone this NetPyNE tutorials directory. Okay. And then, yeah, so we can see it showed up here, the NetPyNE tutorials. We're going to change into that directory, shift enter to CD into it. Because, yeah, a lot of these models have mod files associated with them. So we need to compile those using nrniv modl in the mod dir. So yeah, if we look at the NetPyNE tutorials, all the mod files live in this mod dir. Potassium currents, sodium currents, NMDA, all the different mod files. The cells are all going to be in this directory here. Let's see. So yeah, it's finished compiling the mod files. And again, if anybody needs me to slow down or anything, please raise your hand or speak up. Great. So yeah, so anytime you want to create a NetPyNE simulation, you need your network parameters and your simulation configuration objects. So we're going to shift enter in this cell to set those up. And then I think we already did this, but matplotlib inline allows the plots to show up in the Jupyter notebook. Okay, so the first cell model we're going to look at is a Python file. So we're going to try to import this existing hhcellfile.py. And we can see that here, hhcellfile.py. And yeah, you can see that it's, it imports neuron H, but then it uses Python to create a cell class. And then specifically of that cell class, you can create this HH cell class. And it, creates the geometry, inserts the mechanisms, all those things. And yes, the point here is that, okay, so this is the class. This is what you have to call in Python in order to get an object of this class. So HH cell class with the camel case. We're going to need to remember that. Yes. So yeah, the cell model is defined in a class named HH cell class. So this is under cell name, you have to put that HH cell class so that NetPyNE knows what to call in order to get one of these cell models. Yeah, you have to, put a link to the where the file lives. You can give it whatever label you want. We're going to call this our Python model. And this one has some synaptic mechanisms. So we're going to make that true. So it imports all those. And again, we're just defining dictionaries right now, setting up the configuration for our simulation. We're not actually running anything yet, but this sets it up to import this cell when we're ready to run. Next, we're going to look at importing a HOK file. So neuron is runs in the HOK language, the HOK in HOK. And so, yeah, you'll find a lot of HOK files if you're looking at older models. So this is what a HOK file is going to look like. Begin template E21, it creates a soma and dendrites. This adds a bunch of 3D points to the morphology. So it sits somewhere specific in space. And yeah, so it's a lot of 3D positions for those points. We're going to go way down to the bottom of the file here just to see. Yeah, so the template is called E21. From a Python file, you need the class used to create it. From a HOK file, you need the template name. So going back to here to get this HOK file, we'll label it HOK morphology. There's the file it's going to load from. And then this E21 is the name of the template. So HOK calls this template in order to create an object. So NetPyNE needs to know that. This one it just has geometry. There's no mechanisms, so we don't need to import them. But we will go ahead and add just the standard default Hodgkin-Huxley mechanisms into the morphology, just so it does something. So we'll set a cell rule. The soma is going to get the Hodgkin-Huxley with these values, and then each of them is going to get every section, the soma and the dendrites and everything is going to get the passive mechanisms, and we'll set the membrane capacitance everywhere to 1. So that is, the HOK file. An SWC file is a standard morphology file. It's just a list of 3D points and their connectivity that define a morphology. So by definition, it does not have any mechanisms or anything inside. It's strictly the geometry of a neuron. We can take a look at what an SWC file looks like. So the data looks like this. You've got an index 1, 2, 3, 4. You've got the section type. So 1 is going to be soma, 3 will be dendrite. You got XYZ position, you've got diameter, and then you've got the parent. So the soma, or the very first section of the soma, has no parent. It's by itself. Every other section has to attach to a previous section. So this attaches the first one, that attaches the 2, so on and so forth. And this is just a big line of those 3D points. So to get an SWC file in, we'll just label it SWC morph. Got to point out where the file is. In this case, there is no template, there is no class, it's just a geometry. So you don't need the cell name in this case. There are no synaptic mechanisms, so you don't need that. And again, because it's just the morphology, we're going to do the same thing we did to the Hoke geometry. We're just going to add some Hodgkin-Huxley mechanisms so they're so the cells are active. Now, if when you're importing a morphology, if there's several sections that make up the soma, then it'll be soma 0, soma 1, soma 2, soma 3. But all of our plots, we just want to plot soma. So we actually, there's a function you can call rename cell parameters, and we just want to rename the soma 0 section to soma, so it's easier to deal with in plotting and stuff. Hoke files, a lot of Hoke files you'll find will define a complete cell model, not just the geometry. For example, this PYR3 Traub.Hoke we can take a look at. Oh, this port was made from Fortran into Neuron. That's interesting. But yeah, so it's essentially going to define a template called PYR3. We need to put that in. And yeah, not just a geometry, it does, it's a fairly complex cell model in Hoke, but we can import this easily into NetPyNE, call it our Hoke model, where the file lives. And again, PYR3 is the template name, so NetPyNE can call it to create it. Shift enter in there. And then, yeah, again, we're going to rename compartment 1 to soma, just so that all our plots, we can just point at soma for all of them. Great. So we've got our cell types. Now, in order to get them into NetPyNE model, you have to put them into populations. So we're just going to create 4 populations, each with 1 cell of these 4 different cell models that we're importing. We need to set up a few simulation configuration options. We're going to shorten it to 200 milliseconds and a bigger time step. And we'll record the voltage at the soma for all the cells just to make sure that it's doing something. So we'll set up the record traces at the soma. And then, yeah, we'll set up the traces plots to automatically be generated and saved. So analysis plot traces include all of them, and yes, save them. And then, yeah, we can use our standard single command to create, simulate, and analyze that model. So yeah, I have a network of 4 cell populations on 1 host. Great, here we go. So our Python pop, we can see, all right, it just settles into a resting membrane potential. Our SWC morphology also settles into a resting membrane potential. Ooh, our Hoke model actually gets a little spike before settling in. And yeah, and just to show that we imported the morphologies, we can use this plot shape function to visualize all of these. So you have to include pre and include post. We just want to have each one individually. So we can execute this, and we should be able to see, yeah, here we are. We've got our Python population, our, uh-oh, we lost one. I think that was the SWC and that. I don't know why we lost one. Everything worked fine earlier. I may have missed a command of some sort. But yeah, so it's very easy to get externally defined cell models into NetPyNE. And this is just walks you through a way to do it with several different file types. So yeah, if you have any questions about that, go ahead and raise your hand now. Oh, I see there's something in the chat. It worked okay here. Fantastic. Very cool. Great. So yeah, so now you can, if you find a model you like on Model2D or anywhere, you can get it into NetPyNE and explore it more easily in NetPyNE. So yeah, I'm going to close all these files now. And we are going to go back to NetPyNE tutorials. And that was importing. Okay, so now we're going to go to the NetPyNE analysis plotting Jupyter notebook, and we will explore in a little more detail some of our analysis and our plotting options that you can do. So everybody go ahead and double click on that to open it up. And let's see here, kernel. I'm going to restart the kernel to clear all outputs. There we go, just so it's nice and clean. Okay, so analysis and plotting in NetPyNE should already be installed, but I'll just go ahead and run that command anyway. Pip install Neuron and NetPyNE. And then, yeah, we want to clone our NetPyNE workspace directory. Shift enter on that. And great, we're going to change directory into that NetPyNE workspace. And we are going to compile the mod files with nrniv modl mod. Yeah, there's a bunch of mod files to be compiled. Okay, great, and that's done. Okay, so for this GUI Toot 3 has a lot of different stuff, so it's good for looking at a lot of different analyses. So this is essentially the tutorial 3 from the GUI. And yeah, so we're just going to execute that. And then we can run that simulation with our single command, create, simulate, analyze. All right, and it's got 6 cell populations, 309 connections. It's got reaction diffusion stuff. It's recording 24 traces of 4 types, and it's running the simulation. So yeah, it might take a few seconds here to run that. Yeah, it's taking a minute. Anyways, yeah, once that's done, we're gonna you can call all these things with sim.analysis.plot something, but instead of typing sim.analysis.plot all the time, I'm gonna say SA equals sim.analysis, then I can do SA dot all the plots. And that is still running. Yeah, well, in the meantime, let's see here. How do I add a new cell? There we go. Yeah, it's finished. Okay, yeah, we're done. Okay. So then, yeah, we want to matplotlib inline to make sure we can see them all. We've got SA equals sim.analysis. So now you can hit SA.plot and then hit tab, and that'll show you all the possible tab completions. So you can see very easily what all the plots you can get: 2D firing, 2D net, connectivity, current source densities, dipoles, EEGs, LFPs, the local field potentials, rasters, rates, spectrograms, the morphology with plot shape, the spike histogram, spike stats, traces. So there's a lot of different plots available. Let's go ahead and start by looking at this plot 2D net. And whenever you're wondering what something is, you can always put a question mark after it and then hit shift enter. And so plot 2D net has these options you can put into it: include, view, show cons, pop colors, all these different things. It's got a little doc string here. So include is a list of presynaptic cells to include, and some options. You can do all net stims. You can just do it by population name. Yeah, but for all of our plots, it's set up to with some default values, so you shouldn't have to put in any options to any of our plots. So yeah, just the default plot to DNet. We've got our 6 populations over here, each with a different color, and this is in X, Y space, and you can see where the cells are, and you can see the connectivity between them. And yeah, I think it's a blue connectivity for excitatory. Red and blue are different. I think red is inhibitory, blue is excitatory. And yes. Okay, and so whenever you create a figure, what it returns is going to be a tuple with the figure and its data. So one way to repress that output, the return, is with this semicolon, and then you can see it didn't return what it normally returns. Let's go ahead and take a quick look, though. We can say fig data equals this, and then we don't even need the semicolon. It won't print fig or data. And now let's add a new one. Oops. Plot, yeah, fig and data. So the fig is going to be that. The data should be a dictionary. Yeah. So the data that comes out is the data that's used to plot this. The include, if you're going to include all cells or just populations, and then all the positions of that stuff. So like I said, the include is you can exclude a bunch of different things if you want. So for example, if we wanted to just look at the 3 excitatory populations, we could do include with a list of those populations. And now you can see we've just got E2, E4, and E5. We've excluded the inhibitory ones. Yes, you can change the view. So by default, it's going to come out XY, but you can also view XZ. And YZ and so on. Yeah, so that's the connectivity, the 2D connectivity, 3D connectivity. Now for the overall connectivity, the plot cons, this plots the connectivity matrix. So we've got all of the presynaptic populations, all of the postsynaptic populations, and the average strength between any 2 populations. Again, you can include or not include different things. Let's say we just want to see what excitatory does to inhibitory. So we can do include pre, the excitatory stuff, include post, the inhibitory pops. And then, yeah, you can see the presynaptic side is the excitatory, the postsynaptic side is the inhibitory, and those are the various strengths. And in this connectivity matrix, there's a variety of features you can look at. So by default, the feature looked at is strength, which is a weight times a number of connections. We can also plot just the number of connections between any 2 populations. So from I2 to E2, there's about 20 connections or so. Yeah, you can also group it by cell instead of by population. And in which case we want the feature to be weight instead of strength, because there's only 1 connection between any 2 cells. But yeah, so now you can see all of our 60 presynaptic cells, all of our 60 postsynaptic cells, and these are the connectivities between them and the weights of those connections. Yeah, you can also change the ordering. So normally our ordering is by global ID, by GID. But these are interspersed in space, so maybe you want to order it by Y instead of by population, and it's slightly different. Okay, so then yeah, we can take a look at plot rate spectrogram, which is going to give us the frequencies at any given time during the simulation. And we can see it does it for all cells and then individually for each population. But let's just really zoom in on one of these. We'll just include all cells, where it puts them all into one and analyzes it. And yeah, so this is what, yeah, at 25 hertz, 30 hertz, you can see we've got a lot of power here in this particular simulation. In almost every plot that involves time, you can set this time range. So if you don't want to look from 0 to 1,000, say you only want to go 0 to 400, we can plot that, and now you can see it goes from 0 to 400 instead of 0 to 1,000. Spike histogram is going to show you spiking frequency over time. And yeah, so you've got all your different populations. It gives you some information, you know, the average rate of each population. And yeah, bin size is an interesting option on this one. So if we increase the bin size, then it's, you know, you can see things a little more clearly, see different things. Yeah, plot spike is bin size equals 20. Instead of frequency, we can also do a count, which is just the number of spikes in each time bin. So all cells combined is the black bins, and then each individual population has its color there. Okay, and then there's a variety of spiking statistics you can get out automatically with plot spike stats. And I think this will spit out 4 figures. This first one is going to be the rate for each population, the spiking rate. And these are box plots. I believe the mean is the star, the median is the thick line, and then, you know, second and third quartiles or whatever. But essentially, yeah, you know, this I5 has the highest firing rate here. The second plot is the irregularity. So is it spiking real regularly, or is it real irregular? There's an index you can measure on that. And yeah. So this is where, okay, yeah, there's a couple other spike statistics that we can get out, but we're not going to worry about that right now. This is to show you the fig data comes out of each of these plots I showed you a little earlier. And then plotting traces is one of the most common plots. You know, you're going to record the voltage or the currents or anything. Those are going to all end up being traces. By default, it's going to print a different figure for each cell. And in that figure, it's going to have each trace that it has recorded. So for cell 0, you know, we recorded from the soma, we recorded a couple calciums, and we recorded a potassium current. And you can see the same thing for cell 10. We just recorded from one cell from each population, and we get all those. And yeah, so by default, it's 1 fig per cell, but we can also plot it 1 fig per trace. So if we do that, then this is the soma voltage against all those different cells. And this is the you know, internal calcium concentration versus all those cells. External calcium versus all the cells. If you want to compare these things against each other, then you probably want to set overlay equal to true. And then it'll overlay all those. So this is the voltage soma. These are all the cells in there. The calcium stuff with all the cells all on the same plot. That's the overlay option. You know, there's a lot of things you can do. You can set axis equal to false, which removes the X and Y stuff and adds a scale bar. Yeah. Okay, so traces, raster is also probably our most common plot after traces. By default, you know, you've got your different populations. And you can see they're ordered by GID. They're not ordered by where they actually sit in space. If we want to see where they actually sit in space, we can set order by to Y instead of GID. And that seems odd that they're not interspersed. Normally, I would expect them to be more interspersed now. But yeah, I'm just going to go on. Order inverse is useful for plot raster a lot of times because cortex is usually defined top down, layer 1, layer 2, layer 3, whereas plots usually go 0 up. So if you invert the order here, now you can see that E2, which is on the top of that cortical model, is now on the top of the raster plot by inverting the order. Order inversion labels overlay. Yeah, that's it's an old function. And I'm not sure if that one works anymore. Yeah, ignore that. Sync lines. Okay, yeah. So a lot of this is older options that might not be totally functional anymore. I'm going to have to update this Jupyter notebook. With raster, one thing you can do is you can change the marker. So if you want it to be a circle instead of the little dashes, that's easy to do. And yeah, one other thing is that you can put in custom colors to your populations. So I'm going to create a dictionary called colors with a different color assigned to each of those populations. And then in our plot raster, if we set pop colors equal to colors and execute it, we can see that we get our custom colors into there. And that works on all the other plots as well, this ability to set the population colors yourself. Plot LFP is going to spit out a few figures. So this is going to spit out your time series. So in this model, there were 3 electrodes. So you got the signal from each electrode as well as their average. The power spectral density, which is each electrode, how much power it has at the different frequencies. You got the spectrograms for each electrode. And then you've also got a shape plot of the entire network and where your LFP electrodes sit. And then, plot RxD concentration. This one, you have to put in a species label and a region label because NetPyNE has no way of knowing what you want to plot, so there's no default. But yeah, it's easy to plot the reaction diffusion concentration. And then, last time I also went through sim.sim data to show you where all this stuff is, but Salvador just walked you through all that before my little tutorials here. So questions on plotting and analysis or anything related there? All right, well, I hope that gave you a sort of broad overview of all the different plots and some of the basic options you can use in them.
Great, thank you so much, Joe. That was fantastic. Okay, so we're gonna finish off today's tutorial with the last part, which is parameter exploration. But in this case, instead of using the graphical interface, we're gonna do it programmatically. And so let me share my screen. Yeah. Okay, so for this last part, we're gonna use these tutorials that we downloaded. So the one we're gonna use is this one that says NetPyNE Batch Tutorial 8 Jupyter Notebook. So this is kind of an adaptation that we did from tutorial 8 in the package reference. So if you go here to the NetPyNE website and you go to tutorial 8, it says running batch simulations, and it's purely Python. So we basically adapted this to a Jupyter notebook. So let's just run through this. So the first step is the typical installation of the packages. And then what we're gonna do is basically clone a repository, a GitHub repo that contains the batch tutorial. It requires several files. And so we're just gonna clone that here. This line just removes it in case you downloaded it before, and then we clone it. Okay, tutorial. And then we go to tutorial batch. Okay, yes. So we clone this to this folder called tutorial batch, and then we change directory. So now we're here. Notice that this has several files. It has a config, simconfig and netparams, or config and netparams, and an init file. We haven't really seen much about the structure of these files because we've been running everything together. But typically, the way that models are structured in NetPyNE is that you divide the netparams, so the network parameters, into one of the files, as we see here. This object netparams contains all the cells, the population, synapses, and so on. Then you have a separate file for the configuration, the simulation configuration. And here you see all these typical parameters. And you have a third file, which we call init, which basically just runs the model. So this separation of files is required when you want to run these batch simulations or parameter explorations. Notice that we call batch simulations when you want to run more than one simulation at a time, and that is what is required to explore parameters. So yeah, so in this model, it's already separated into these three main files. And then here we have two other files. This batch, which is the one that contains the setup of the parameter exploration, which parameters you want to explore and how to run it. And finally, one that it's analysis, which is kind of optional, and it just reads the data from the saved files and does a nice plot. This is really custom code. It's not really part of NetPyNE, but just a nice way of visualizing the output together. So what we've done in this tutorial is just move these two files, the batch and the analysis, make it into a Jupyter notebook. And that's what we're seeing here. You see one of the cells here is this setup batch, which specifies the parameters and so on. And then the next cell is just the analysis that we pasted here. Okay. And this is just, again, optional, not really part of NetPyNE, it's just Python code. Okay. So I'm going to remove the two Python files here just to avoid confusion, because these are part of the Jupyter notebook now. Okay. And so essentially what you have remaining is this setup of the batch and then some analysis to plot the results. And we'll see how this works. So when you want to set up a batch, you have to import this netpyne.batch batch object or batch class. And then you define the list of parameters that you want to explore. So in this case, we're going to explore two of them. And when you have two, then you are going to have the combinations of all parameters. So in total, we're going to have 4 simulations exploring the 4 combinations of parameters, 2 times 2. So the 2 parameters we're going to explore are the synaptic mechanism tau2 and con weight. Well, you might be wondering where these are defined, because these are just like simple labels. And this is where the setup comes. So you have to go to the tutorial 8 config file, so this simulation configuration file, and you have to set up initial values here for these 2 parameters that we want to explore. Synmec tau2, we're going to set at 5, and con weight at 0.01. And then in netparams, which is the file where we define the network parameters, here we're going to use these variables defined in simconfig. And so this might seem a bit convoluted compared to how easy it was to set up in the graphical interface. But essentially what we're doing is sort of parameterizing some of the parameters in netparams. In this case, these 2, synmec tau2 and con weight. And these are defined inside simconfig and then through this batch, we change, or NetPyNE will change the different values and run. So from your perspective, when you want to set up this type of batches, the first thing you need to do is go to your netparams, decide which parameters you want to explore, and replace them with some variable that is part of this config object. And then you have to define initial values or baseline values in simconfig. And finally, you can just set, using the same label that you have in the simconfig, set the values that you want to explore. The next step is to create this batch object, where you pass as arguments first the parameters and the values that you want to explore. And then you have to say which NetPyNE files you're gonna use, which config file, which in this case it's called tut8config, and which netparams file, which is called tut8netparams. And finally, you just need to set some configuration options for this batch. For example, the label of this batch, which is going to be tau weight, that's the name of the two parameters, the folder where you want to store the output, tutorial 8 data, we called it, and the method. Notice here the method we're using grid, which stands for grid search, where you just give fixed values for each parameter. But we also have other methods like evolve or evolutionary algorithm, and other actually more machine learning based methods. So you can read about this in the NetPyNE documentation. And the last set of options you need to set is the run configuration. So how are we going to run this set of simulations to explore parameters? And in this case, we're going to use something called NPI bulletin. This is a method that is built into Neuron. It's called the bulletin board. And it essentially creates a master node or a master core that sends jobs to the slave nodes. It's a feature that is built into Neuron, so we made use of it here in NetPyNE for this purpose. And you select it using this label, NPI bulletin. So it's going to use NPI and the bulletin board of Neuron. You have other options like NPI direct, where you just run using whatever NPI resources are available. And you can also use, for example, Learn, which is a workload manager for supercomputers. So you have examples of all this in the documentation and within the examples folder in NetPyNE. And next we set which is the script that you're gonna run. So the initial file that you're gonna run to run the simulation. And this skip through is just an option to say that if the output already exists, you can skip that simulation. Okay, so this is the way we set up this parameter exploration using batch simulations. And the final command is just b.run. B is the batch object, and we tell it to run the simulation. So let's go ahead and run this cell here. And what you see that happens is that NetPyNE is in the background running the different simulations and with the different parameters. So you can see here 10 and 0.05 are the first pair of values that it's running. Then 10 and 0.01, which are this and this, and so on. And saving the output of each simulation to a different file or to a different set of files. Okay. And we can look at that here. This folder, we can also delete. Yeah, that's just double. Oh, well, anyway, we can leave it there, but it's just, it cloned it again in the wrong place. So we don't really need this one. But the output of the simulation is going to be in this tutorial 8 data. Oh, it's doing everything inside here. Okay, so it's I just cloned it in the wrong place, so everything is duplicated one inside the other. Sorry about that. But basically, you can see here the output of this batch simulation is inside this folder, tutorial 8 data. It was generated a minute ago because I ran the simulation, and you can see for the different parameter combinations, the output of the simulation. If you click on the raster, this is for the first set of parameters. And if you click here, you will see a different raster for different values of the parameters. Okay. So this was all run automatically by NetPyNE in the background, and you got 4 different simulations. Okay. And finally, this is, as I mentioned, some Python code just to load all this data and do a nice combined plot where we're seeing here the 2 parameters that were explored, which is the synmech tau2, one of the synaptic mechanisms tau time constants, and the weight of the connectivity of the connection. So you can see in a very intuitive way, the color here represents the firing rate of the M population, and you can see how these 2 parameters affects the firing rate of the M population. These higher, lighter values represent higher firing rate. So now you can make an informed decision about what parameter values you want to use in your model, or what are the effects of these parameter values, and so on. Okay, so this was just a quick overview to give you a flavor of how you would set up this kind of parameter explorations using directly scripts. As I mentioned, there are more examples, and you can find all the resources here at the beginning of the slides. Here, slide 36 has a list of the NetPyNE resources with tutorials and also example models and so on that you can have a look at. And I think that's it for today. So please let me know again if you have any questions. Okay. Thank you. Excellent tutorial. Thank you so much. If not yet, thank you everyone who attended. I know it was a long one. If you have any questions or you're interested in using NetPyNE for your models, please don't doubt to reach out to us either by email or through our forums. We're always happy to help users, and it's actually part of our funding to help people. And yeah, thank you so much, and hope to see you soon again or stay in contact with you. Thanks a lot. Yes, so one of the questions in the chat asked if we will make the video available, and yes, we're going to upload the video of the 2 sessions to YouTube, and we will post the links on the different social media and on LinkedIn, on Twitter, and also we will mail it through the NetPyNE mailing list. Let me just say we have a mailing list if you're interested in signing up with all the information. So this is netpyne.org/mailing. Here we will, oh, wait. Let me message this to everyone. We will post the videos here as well, but you can also follow us on Twitter. Twitter is NetPyNE_. Okay, thanks again, everyone.